Hello students, myself Prokaryotes and you are welcome to Biology Reward. Today we are discussing class 12th chapter molecular basis of inheritance, and our topic is the process of transcription in prokaryotes and eukaryotes. The process of transcription in prokaryotes and eukaryotes. So, in previous class, we have discussed about the transcript unit. In this transcript unit, the molecule of DNA, one strand of the molecule of DNA, it transcript into the RNA. It means it convert into the RNA. So, when DNA transcript into the RNA, this is called the transcription. Now, we have to look at the transcription process in prokaryotes and eukaryotes. So, how the transcription occur in prokaryotes and eukaryotes, and how the transcription process is different in prokaryotes and eukaryotes. So, this we have to discuss now. Students, as we have learned, the gene is a segment of the DNA. A gene is a segment of DNA, and this segment of the DNA it determines the characteristic. Here is another gene which is called cistron. So cistron is a segment of the DNA, and this segment of the DNA it are coding for polypeptides. So that segment of the DNA, or it is one gene which code for polypeptide. So this gene is called the cistron. So what is cistron? Cistron is segment of DNA. It's coding for polypeptide. Polypeptide chain is formed, and by the polypeptide chain, the protein molecule will be formed. It means a gene which is responsible for synthesis of the protein is called the cistronic gene. So cistron is a segment of DNA. It's code for polypeptide. Second, transcription in prokaryotic we have to discuss. So this cistron is a segment of DNA code for polypeptide. It's occurring both prokaryotics also and eukaryotes. So we have to discuss now the transcription in prokaryotes. So in transcription, first point is there is a segment of the DNA which code for polypeptide, and it is called the cistron. So what is cistron? It is one gene, segment of DNA code for polypeptide. Number 2. In prokaryote, the structure gene is polycistronic. It means transcription process, many such cistrons are used. Many cistron, polycistronic. So prokaryotic transcription process is regulated by the many cistron, and that's we are saying structure gene is polycistronic in prokaryote. Polycistronic means many cistrons. Then third is in transcription process in prokaryotic, DNA dependent RNA polymerase, the very important RNA polymerase enzyme. It catalyzes the transcription of all types of RNA in bacteria and prokaryotes. Try to understand transcription process. One enzyme is required, and it is RNA polymerase. So this RNA polymerase enzyme, where DNA molecule it transcript to form the RNA. So for the formation of the RNA, it's required RNA polymerase enzyme. So RNA polymerase enzyme is required for transcription of RNA. RNA are different types. It may be mRNA, rRNA, tRNA. So all this type of RNA are synthesized or formed only by RNA polymerase enzyme. So first point, cistron is held for polypeptide chain. Structure gene is polycistronic here, many genes are, and RNA polymerase is held for the transcription process, and this RNA polymerase form the different type of RNA. rRNA, mRNA and tRNA. Now, how this transcription process occur? What are the steps of transcription process in prokaryotic? So, students, we have the important three steps for the transcription in prokaryotic. One is called initiation. First step is initiation. Second, elongation. And third, termination. Initiation, it means simply you understand the process of transcription is started. Elongation means process of transcription will be continued throughout the DNA molecule, and one strand of the DNA continuously will form the RNA molecule. That is called elongation. And third, where the transcription process is blocked, transcription process is stopped. Okay, so this is called termination. Now let's we understand first process, it's the initiation. What is initiation? It's a process in which RNA polymerase, this enzyme, RNA polymerase bind to promoter, it means fifth end, associated with initiation factor, it's a sigma factor. Three important things we should understand. RNA polymerase bind to the promoter. You will see this is the DNA strand. In DNA, there one strand is 5 to 3rd polarity, other is having the 3 to 5 polarity. So promoter gene in transcript unit we have learned, the promoter gene is located into the 5th site. So if promoter gene is located to the 5th site, so this is the site where promoter is located. So what happened? In this site, this promote RNA polymerase enzyme. In promoter site, the RNA polymerase enzyme, this is the name polymerase enzyme I have made. So this RNA polymerase enzyme, this enzyme, white color I have indicated, it is the RNA polymerase enzyme. This RNA polymerase enzyme along with this one is a sigma factor, or we are saying initiation factor. Sigma factor. And this initiation. So RNA polymerase along with the sigma factor, it bind with the promoter site. It means in 5th end, here the promoter site is RNA polymerase is bind. So when RNA polymerase along with the sigma factor is bind with the promoter 5th end, promoter site of the 5th end, and this process of transcription is begin, and that is called initiation. So what is initiation? It is the process in which RNA polymerase along with the sigma factor is bind with the 5th end of the promoter site of DNA molecule. When this process is begin, it is called the initiation. After initiation, what happens? This RNA polymerase will start the uncoiling of molecule of DNA. And out of this one, 2 strands of DNA, only one template strand will transcript into RNA. So here is the RNA polymerase location you will see. And second diagram you will see here position of the RNA polymerase is different. It means this complete strand of DNA is changing to RNA. And throughout the DNA molecule, this polymerase enzyme transcript the DNA to RNA. So this process is called the elongation. What is elongation? The DNA strand uncoiled and RNA polymerase, it synthesizes the RNA strand by the transcription of the DNA. So out of 2 strands of DNA, one DNA strand is transcribed to RNA, and this occurs throughout the DNA molecule. And this process is called elongation. And third process is termination. In termination, what happens? Here you will see this DNA molecule is continuously forming, and this is the RNA. Now RNA polymerase here, it reaches to the last point here, and here RNA polymerase when reach to this point, there is a one factor. It is called terminator factor, and it is also called rho factor, P rho factor. So when the RNA polymerase comes in the site where what is there? Terminator factor is there. Here you will see some nonsense codon also comes. So that nonsense codon along with the terminal factor comes in the site. And when this terminal factor comes, here RNA polymerase stop working. And if RNA polymerase stop working, the transcription process stopped. So when transcription process is stopped, when the terminal factor comes here, and this terminal factor, it stops the transcription of the RNA. So RNA polymerase working is stopped. So here the RNA formation process is stopped, and this is called termination. So first diagram show the initiation process where RNA polymerase here. Second diagram is of the elongation, and third is of the termination. So here you see RNA molecule is formed. So when the process is terminated or stopped, this RNA molecule, this will switch off. It means it fall off. This RNA molecule will be separated. It means one strand of DNA is transcribed or changed into RNA. So overall you will see in transcription process through this diagram also. In transcription there are three processes. One, initiation process. In initiation process, RNA polymerase along with the sigma factor is combined with the promoter site, hit the end, and the process of transcription begin. It means the DNA molecule one strand start to transcript to RNA. Second, elongation throughout the DNA molecule, RNA polymerase uncoil the DNA and one DNA strand is formed. That is called the elongation. And when rho factor comes, terminator factors come along with nonsense codon. So here RNA polymerase, it stop working. So process of transcription is terminated. It is stopped, and this is called the termination. So in this diagram also the prokaryotic transcription is exhibited. So we should remember prokaryotic transcription, initiation, elongation and termination. Now if we compare the transcription with the eukaryotic. So in eukaryotics also, the process of transcription are similar. Process of transcription in eukaryotics also, here I have mentioned only steps. Initiation, prokaryotic, elongation, prokaryotic and termination. So like the prokaryotic transcription, the same process are occurring eukaryotes. So eukaryotes also we are having three steps of the transcription: initiation, elongation and termination. What happened in elongation in the eukaryotic also? Same thing will come. Here RNA polymerase will bind. Now we have to discuss now what are the differences in the transcription process in the eukaryotic. Let's know. Transcription in eukaryotes. First one, as I told you, segment of DNA coding for polypeptide. What it is called? Cistron. Here also cistron is there. But in transcription, here cistron structure is monocistronic. Transcription process, here many cistrons are there which regulate in prokaryotic. But in eukaryotic, there is single cistron. Single cistron regulate the transcription. Second, monocistronic structure gene have exon and intron. Look at here, what are these? Monocistronic single gene is involved in the transcription, and this monocistronic gene is having exon and intron. There is a interruption of two segments. Two different type of segments are there. One exon and one intron. Where? In cistron. In cistron, there are two type of sequences. One is exon and another, and they are interrupting to each other. Now, what is here? You will see this is the molecule of RNA I have made. So in molecule of RNA, monocistronic structure, this green color structure, they are the intron. and red color of structure I have mentioned as the axon. So in monocistronic gene there are two type of structure or sequences, and one structure genes, axon and intron. Axon, this is the axon, and axon is what? Axon are the coding sequences appear in the RNA. Transcription process what happen? DNA transcript to RNA. So that sequence which occurs in the RNA, it means axon are the coding sequence appear in the RNA. So here you will see these molecules of RNA. In these molecules of RNA, only red color segments are present. And what are these? Axon. So axons present in the RNA, and that's we are saying axon are the coding sequence appear in the mRNA. Now this appear in the RNA. So RNA formation take place. RNA formation take place by the axon. So many time question is asked, what is axon? So we should write axon are coding sequences which appear in RNA. So for second sequences are also there. These are called non-coding sequences. And these non-coding sequences not appear in RNA. You will see last one end product which RNA form, it do not have this intron. It means the introns are non-coding sequences not appear in RNA. So what is axon and what is intron? So monocistronic gene, there are the two type of sequences. Axon are the coding sequences which occurs in the RNA, whereas introns are non-coding sequences not appear in the mature RNA. So this is the different than the prokaryotic. Prokaryotic are polycistronic, this is monocistronic, and this monocistronic also axon and introns are present. Third important thing which is differ, that is RNA polymerase. In transcription process in eukaryotic, here 3 RNA polymerase involved. Here was only single RNA polymerase and it synthesized the all different type of RNA. But here 3 RNA polymerase are there. So 3 RNA polymerase involved in the transcription. This is the another difference. 3 polymerase enzyme. First one, RNA polymerase 1. What is the function? It form the rRNA, it means transcript the rRNA. RNA polymerase first, it form rRNA. Later we will discuss rRNA. So it form the rRNA or transcript the rRNA. 20S and 18S, this type of RNA. RNA polymerase third, this transcript the tRNA. It means it help to formation of tRNA, 5S rRNA and snRNAs. It means small nuclear RNA. So RNA polymerase third, it form the tRNA, 5S RNA and snRNA are small nuclear RNA. And another RNA polymerase second. This enzyme, it transcript the precursor of mRNA. mRNA formation take place and hnRNA, it means heterogeneous nuclear RNA. So here you remember, RNA polymerase is single and it form the all type of RNA in prokaryotic. But here there are 3 type of RNA polymerase in eukaryotic, and RNA polymerase 1, it form the rRNA. RNA polymerase third, it form the tRNAs, 5S RNAs, snRNAs. And RNA polymerase second, it form the transcript precursor of mRNA. So initial part, mRNA is formed, that is called precursor of mRNA. Later the mature mRNA is formed. So it is called transcript precursor of mRNA and hnRNA. These two are formed by the RNA polymerase second. So this is the very important part, function of RNA polymerase 1, RNA polymerase third and RNA polymerase second. The sequences I have written according to your NCERT book only. So RNA polymerase 1 function, RNA polymerase third function, RNA polymerase second function. So this is the one more different. Third, as I told you, it is monocistronic gene. In monocistronic genes, what happens? Monocistronic gene, there are this DNA molecule. Look at it. In monocistronic gene, there are exon and intron. Here you will see, children, this diagram. It is the molecule of DNA. In molecular DNA, now first process will start, initiation. What happened here? This RNA polymerase along with the sigma factor like the prokaryotic. So this RNA polymerase along with the sigma factor combined with the promoter site here. So when it combined with the promoter site, the process is started. It is called initiation. Second diagram, this RNA polymerase will uncoil the DNA and throughout the DNA is change the DNA strand to RNA strand, and this process is called the elongation. So here is RNA polymerase and sigma factor. So position was earlier here, but now the RNA is formed here. And last process I have discussed, termination. What happened here? In this process, the Rho factor will stop the functioning of RNA polymerase. And last one, what we are getting? We are getting this RNA. Now this RNA is synthesized 5 to 3 direction. It means template stand is 3 to 5. So this 3 template stand, 3 to 5 stand, this is converted. It is 3 to 5th polarity DNA stand is converted into RNA. So synthesis of RNA take place in 5 to 3rd direction. Now this RNA is formed. 5 to 3rd direction RNA. Now to see in RNA there are 2 types of band. One band I have mentioned exon and another bands are the intron. So this yellow color band, it is at the intron. This yellow color are the intron. Intron. And red color, as I have mentioned here, red color are the exon. Okay. So when RNA molecule is formed from the DNA, it means DNA is get transcribed to RNA. Then this mRNA like of, or this is initial RNA we are saying, and it is called the precursor of the mRNA. Now the fully mRNA molecule is not formed, so it is said the RNA is formed. This RNA is a precursor of the mRNA. So first one thing is formed, this is the RNA and this is precursor of the RNA. Once the RNA is formed, now what happens? Or how this RNA formation takes place, here you see. RNA splicing is a one process. This RNA molecule is formed and here intron and exon are both present. Now what happens? RNA splicing. What is RNA splicing? Introns are removed and exons are joined. So out of this RNA molecule, the intron sequences are removed and exon sequences are joined together. Okay. So this process is called RNA splicing. RNA splicing is what? Removal of intron and joining of exon. Okay. So all the exon segments which are away from each other, after the removal of intron, they will join together, and this is called the joining of the exon. To more process, when the exon are joined only, exon is formed. Exon segments are formed. Now what happens when the exons are here only, completely exon? Now other processes started in eukaryotic. And what is this? Capping. When the RNA molecule is formed, this is the 5th end. So in 5th end, methylene guanine triphosphate is added. This is the RNA molecule. In this RNA molecule, I told you precursor RNA, precursor of mRNA. So in this RNA molecule, introns are removed and exon are joined. So in 5th end, here methylene guanine triphosphate is added. So this process of adding the methylene guanine triphosphate to the fifth end of the heteronuclease RNA. So on the fifth end, what is added? Adenylene guanine triphosphate, and this process is called capping. So I have made the capping. So capping takes place, it means on fifth end methylene guanine triphosphate is added. Second, tailing. On third end of RNA, what happens? Adenylate residues, about 200 to 300, the adenylate residues are added. At the third end of this is called heteronuclear RNA. So on third end, what is added? Adenylate residues are added. So it is polyadenylate tail. So this process is called the tailing. Capping, adding of methyl guanine triphosphate on fifth end, that is called capping, and adenylate residues are added in the third end of the RNA, it is called the tailing. And after capping and tailing, now it was the precursor of mRNA. Now we are getting the last one point. Capping is also here. It means methyl guanine cap is there. Tailing is also here. Here the adenylate residues is there. And all the exons are joined. It means introns are removed. Now this molecule is formed. It is called mature mRNA molecule. So in this all process from DNA, it transcribes to form the mRNA molecule. In this diagram also you can see eukaryotes. In eukaryotic DNA, RNA polymerase, it combining with the promoter site. When it combines, the process of transcription is begin. When process of transcription begin, then RNA polymerase throughout the molecule of DNA, it synthesizes the RNA molecule. So when RNA molecule is formed, in this process, the 3 RNA polymerase are used. So when RNA molecule is formed, that is called the precursor of mRNA. And after the formation of precursor of mRNA, introns are removed and exon are joined, and that's precursor mRNA is formed. After that, methylene guanine triphosphate is added in the fifth end, that is called the capping, as well as adenylate residues is added in the third end, that is called the tailing. And after this process, we are getting the mature mRNA, and this mature RNA is called messenger RNA. mRNA stands for messenger RNA. It means DNA one strand, it transcribes to RNA. Now overall, what are the differences? Differences is in prokaryotic the cistron is polycistronic, but in eukaryotic it is monocistronic. In monocistronic there are exon and intron. Here exon and intron are not there. Third, here RNA polymerase is only required for synthesis of rRNA, but in eukaryotic 3 types of RNA polymerase are required. Third, RNA splicing occur in eukaryotic. It does not take place here in prokaryotic. Capping and tailing is also occur in the eukaryotic. So here this process is not described here. So, and process and steps of the transcription, initiation, elongation and termination, these are the same in the prokaryotic and eukaryotic. So it is all about the transcription in prokaryotic and eukaryotic. Thank you.